All right, welcome back from spring break. Before we jump into chapter 24, I just want to say a little bit about the new lecture system. I had a few people reach out to me and say, please don't make us do PowerPoints. Do the whiteboard as much as you can. And so I'm going to try to do that. I haven't changed my lecture notes at all. But one thing that's going to be different is that I'm not going to be able to jump back and forth between the whiteboard and PowerPoint like I normally like to do. But since we're using a video, right, you're watching this on a video, at any time you want, you can hit pause and then look at the PowerPoint. And if you're on a laptop, you can have both of these open concurrently, and you can have my video open, and then in a separate tab, you've got the PowerPoint open. So if I start talking about the PowerPoint, you can just minimize the video, listen to my voice, and look at the PowerPoint. So, and if you see me walk over here off screen, I'm going over to the computer over here so I can advance the slide, because that's what I'm looking at. That's my monitor. So that's what happens if I walk over there. I'm going to try to speak really loudly because my camera's not very good. So feel free to let me know if I'm being too quiet, and I'll try to fix that for subsequent videos. And hopefully you've all got a laptop or a desktop computer. Because if you're watching this on your phone, you may notice, like, that's really hard to read. If you're looking at this on a phone, you might not be able to read what I'm writing down. If you don't have a laptop or a desktop computer, shoot me an email, and we have ways around this. We'll do what we got to do. But hopefully, I think, it's year 2020, hopefully all of you have some access to some kind of computer. So hopefully that's not going to be an issue. But if it is, just let me know and we'll fix it. Yeah, so I'm going to try and post all these videos as quickly as I can make them, because I'm not going to be allowed in here after March 30th, I think. So our first day back, like I can't come back in here. So I'm going to try to bang this out real quick. So, chapter 24 is all about the urinary system. And I guess we'll start off with our PowerPoint. So if you look at slide number 1, well, I guess technically slide number 2, the first content slide is just a gross overview of the urinary system. And so what you can see in the image on the right is that most of the elements of the urinary system don't actually sit inside the abdominal cavity, right? They're in that area, right, between here and here, but they're not within the abdominal cavity. They're actually kind of behind the abdominal cavity. So you can look and see the peritoneum actually passes in front of the kidneys. And so they sit behind the peritoneum. That's why they're called retroperitoneal, because they sit behind the peritoneum, not within the peritoneal cavity. And so that's why if you ever get a kidney removed for whatever reason, hopefully you don't, right? Or if they have to do some kind of surgery on your kidney, they always open you up from the back. They'll open you up from back here, because they actually sit behind the abdominal cavity. The right one, you can tell from the picture, the right one sits a little bit lower than the left one. They don't sit at the same level. The right's a little bit lower than the left. And yeah, all those elements from the kidney on down to the bladder just sit kind of behind, and then the bladder sits underneath the peritoneal cavity. So none of it's actually within the abdominal cavity. Okay, So we will start with just the gross structure of a kidney. So here's our kidney. There you go. There's your kidney. There is a tube that extends off this side of the kidney called the ureter. This is the tube that carries urine from the kidney down into the urinary bladder. And also right here at this indentation called the hilum, this is where the blood vessels enter and leave the kidney. So you have the renal artery and the renal vein. So the renal artery and the renal vein both enter and leave the kidney through here. Like a lot of organs, the kidney has an outer area that is functionally and histologically discrete from the inner area. So there's an outer cortex and an inner medulla, just like you see in a lot of different organs that we've looked at. So you've got an outer cortex, and then inside the dotted line is the medulla. And within the medulla, there's a lot of interesting organization going on inside the medulla. Oops. In the medulla, you have a whole bunch of these structures called renal pyramids. And they're called renal pyramids because they're actually kind of shaped like a pyramid. Well, they're more cone-shaped, I think, right? So they're cone-shaped with the pointy bit pointing in towards the medulla and then flat bit pointing out towards the cortex. So we'll just draw 4 renal pyramids in ours. So this is a renal pyramid. And there are extensions of tissue that they look like they're extending from the cortex down between the renal pyramids, but they extend down into the medulla. Those are called renal columns. So these renal columns are just extensions they look like extensions of the cortex tissue just projecting down between the pyramids. And let's see, wait, what was it at? Here, I'll fill this in. So if you follow the ureter back into the center of a kidney, you'll end up in this open space. It's called a pelvis, right? So this big open area in here is a pelvis. And you can follow from the pelvis. It's like a little labyrinth of tubes that go back into these areas at the tip of the renal pyramids. These little enclosed areas right here are called calyces. I'll write all this down for you. So here in the center is the pelvis. And then these areas, put a star because I can't write in there. It's called a calyx. So here's a calyx right here, here's a calyx right here, here's a calyx here, and a calyx here. And, you know, just very, very broadly, the way this organ functions is urine will be produced by these renal pyramids, and then it will travel from the renal pyramid down into a calyx, and from the calyx it will move on into the pelvis, and then from the pelvis it will move on down into the ureter. So in red, I'm going to draw urine flow. So you got urine being produced by this renal pyramid, goes into this calyx and on into the pelvis. And again by this pyramid. And again by this pyramid. The images in your book are a lot cleaner than this. I mean, I can hardly do it justice. If you look at the pictures in the book, it'll clarify all of my really bad handwriting. Normally I do much better, you can see. Here's the one in my notes. If you want to take a screenshot of that. There you go. All right, so that's just a broad overview of the structure of a kidney. What's next? If we zoom in, we're going to zoom in right well, I'm going to roll today. Sorry, I'm out of practice. It's been a long week. We're going to zoom in right there. Right? What we're going to do now is we're going to look at the border between the cortex and the medulla, specifically the top of a pyramid, and we're going to look at the functional unit of a kidney. It's called a nephron. So we're looking at the border between the cortex and the medulla. So up here we've got our cortex, and down here is our medulla. So above the line is cortex, below the line is medulla. So we'll look at the functional unit of a kidney. It's called a nephron, and it starts with a little kind of bulbous structure called a renal corpuscle. A renal corpuscle. And the renal corpuscle has it's fed by a blood vessel called the afferent arteriole, and then blood leaves it through a blood vessel called the efferent arteriole. We'll draw that. So you got an afferent arteriole and then an efferent arteriole. So the afferent arteriole brings blood to the renal corpuscle, and then the blood leaves via the efferent arteriole. And basically what's happening is fluid gets pushed out of the blood into this renal corpuscle, and then from there the fluid goes into a very convoluted series of ducts called the proximal convoluted tubule. So we'll just draw it like this. It's actually way more convoluted than that, but we're just going to draw it like that. All right, so there is our proximal convoluted tubule. From there, the fluid enters a duct that extends deep down into the medulla and then turns around and comes back up. This is called the loop of Henle. A fellow named Henle found it and named it after himself. The loop of Henle. Right? And then after that, the fluid enters another convoluted tubule called the distal convoluted tubule. And then from there, the fluid is collected into a duct called a collecting duct. And the collecting duct can collect fluid from multiple distal convoluted tubules. So if you look at a collecting duct, it will have multiple distal convoluted tubules emptying into it. So this right here, that's a nephron. This whole thing from the renal corpuscle all the way to the collecting duct is called a nephron. And this is what we call the functional unit of a kidney. You know, if you want to understand what's going on in a kidney, you know, just distill it down into its most simple form, then what you're looking at is basically a nephron, which we'll go into a lot more detail about what's happening here. But so when you look at the cortex of the kidney, there are just thousands and thousands and thousands of nephrons, and the sum of all of their activity is what, you know, constitutes kidney function. And so, you know, you'll have multiple nephrons emptying into one collecting duct. You have far more nephrons than you have collecting ducts, because each collecting duct collects urine from many different nephrons. The proximal and distal convoluted tubule, these names are easy to remember because, you know, proximal just means close to. So the proximal convoluted tubule is the one that's close to the corpuscle, the one that's close to the origin of the structure, which is the corpuscle. And then the distal convoluted tubule is the one that's farther away from the corpuscle. So that's just how they got their names. So talk just a little bit about what's going on in each section of the nephron. Right? You've got the corpuscle. Then you've got the proximal convoluted tubule. Then you've got the loop of Henle. And then you've got the distal convoluted tubule. One of the nice things here is I don't have to ask if I can race anymore, because if I get ahead of you, you can just hit pause and then catch up, and I can just blow through this. So you got corpuscle, which is where the filtration happens. Filtration of what? Blood, right? You have blood entering the corpuscle, and it's filtered, and a lot of the fluid gets pushed out, which, we'll go into a little bit more detail about that in a minute. But that's what's happening at the corpuscle. The corpuscle produces this filtrate of blood that then passes into the proximal convoluted tubule. And in the proximal convoluted tubule, things are reabsorbed out of that filtrate back into the blood. But again, we'll go into more detail about all of this. I'm just giving you big picture stuff right now. So in the proximal convoluted tubule, that's where things are reabsorbed from that filtrate back into the blood. In the loop of Henle, it all gets concentrated. And in the distal convoluted tubule, you get more reabsorption. Right? So in both the convoluted tubules, solutes are being reabsorbed out of the filtrate back into the blood, and then the loop of Henle is where it's all concentrated. So the way a kidney works, the way a nephron works, broadly, what happens is, the blood gets filtered out at the renal corpuscle, and basically everything gets pushed out of the blood, everything except for cells and big proteins, that stays behind, but everything else just gets dumped out of the blood into the filtrate that then goes on to the proximal convoluted tubule. And then in the proximal and distal convoluted tubules, the cells that make up that epithelium kind of selectively pick what they want to take back up out of the filtrate and put into the blood. So it's one way for your body to just push everything out of the blood and then only reabsorb the things that need to be reabsorbed. So bear that in mind when you're looking at how a nephron works and what's actually happening, right? You start with blood, all the fluid gets pushed out, all the solutes get pushed out, except for proteins and cells, and then the important solutes get picked back up, and then everything gets concentrated, and then we pick up some more important solutes, and then everything gets eliminated. Everything that didn't get reabsorbed gets eliminated in the urine. So, we'll start with the renal corpuscle. What's going on there? The renal corpuscle has two parts. One is Bowman's capsule, which is basically it's a simple squamous epithelial layer that lines the outside of the corpuscle, because it's like this small little pocket within the renal cortex. So that's Bowman's capsule. And then there's a tuft of capillaries within the capsule called a glomerulus. And I'll write that bigger, I'm sorry. A glomerulus. So we'll draw that. So here's our Bowman's capsule. All right, what I'm drawing is just a simple squamous epithelial layer. We're going to leave it open at the top for now. Bowman's capsule. Simple squamous epithelium. And then you've got those blood vessels entering and leaving. You've got an afferent arteriole and then an efferent arteriole. But once the afferent arteriole, I'll just do that, afferent arteriole. When the afferent arteriole enters Bowman's capsule, it opens up into this very convoluted, heavily fenestrated capillary bed called a glomerulus. All right, so there's our glomerulus. And like I said, it is heavily fenestrated, very heavily fenestrated. So that means there's a lot of holes in this thing, lots of big intracellular clefts. So when the blood enters this glomerulus, it gets pushed out. A lot of it gets pushed out into this capsular space right here, which I'll write that. Here's our capsular space, and the blood just gets pushed out of this glomerulus into the capsular space. Not the blood getting pushed out, but the fluid within the blood, right? The plasma gets pushed out into this capsular space. All the cells stay behind, all the proteins stay behind. But because the capillary bed here is so heavily fenestrated and thin and very porous, the structural integrity of the thing is, it's pretty fragile. So there's another population of cells here within the capsule called podocytes. And they're called podocytes, P-O-D-O, podocytes, because they have these little feet-looking things that wrap around the capillary bed and just kind of hold it together, right? If we look closer at one of these podocytes, I'll draw, here's our capillary. Podocyte will kind of sit on it like that, and it has these little feet-looking things that extend out and wrap around it. And then between those feet, it has even more cytoplasmic extensions that form this kind of network. It's like a mesh that surrounds the capillary and kind of holds it together. And I'll show you a picture. You might have already looked ahead on the PowerPoint, and you might have already seen that the next picture on the PowerPoint, I think, I'm guessing here. Is it? Yes, it is. It's podocytes. All right, so the next PowerPoint slide, on the left you've got you got an electron microscope image of a few podocytes that are surrounding that glomerulus, and you can really see where they have these little feet that wrap around the capillary bed, and then those little intermeshing little cytoplasmic extensions that surround, that kind of hold the feet together. So that's what you're looking at on the left. And if you look on the right, what you're looking at, you might recognize this from when you were studying for A&P 1, because I like to use kidneys as an example of simple squamous epithelium. What you're looking at is a glomerulus. So you can see that tuft of capillaries within the renal corpuscle. That's the glomerulus, and then on the bottom right-hand corner of that image, you can see the afferent arterial, actually that's mislabeled. Isn't that nice? Wait, no it's not. No, it's not mislabeled. That bottom right corner is the afferent arterial. The proximal convoluted tubule comes off the top left corner. Yeah, okay, no, they got it right. Yeah, so there are a lot of podocytes surrounding these capillaries, holding them together. And you can even look at this and you can see how the podocytes actually contribute to filtering the fluid that's getting pushed out of the capillaries. Because anything that gets pushed out of that capillary, not only does it have to cross through the capillary endothelium, it's also got to cross, it's got to pass between the feet of that podocyte. So the podocyte feet kind of work to hold in all the stuff that shouldn't be leaving the capillary bed. Yeah, and so once the fluid gets pushed out of the glomerulus, then it enters into the proximal convoluted tubule, which is lined with simple cuboidal epithelium. So here's our proximal convoluted tubule right there. So what exactly gets filtered out? I've touched on this, but we haven't really focused on what's actually being filtered out. If you look at all of the nephrons in both of your kidneys throughout an entire day, the amount of fluid that's pushed out of all of those glomeruli together amounts to about 180 liters per day, which is a lot, right? 100, and can't talk and write at the same time, 180 liters per day filtered out by the renal corpuscle. So that's what gets pushed out of the glomerulus. That's the amount of fluid that gets pushed out of the glomerulus, and that's what goes into the proximal convoluted tubule. But you only urinate about 1 to 2 liters per day of urine. I don't know, depending on how well you hydrate, right? Some people less, some people more. Just drink more water, usually, right? But unless something's really wrong, you're not peeing 180 liters per day. So this is what gets filtered out, right? But throughout the rest of the nephron, all but 1 or 2 liters gets reabsorbed back into your blood. So let's talk a little bit about what actually gets pushed out of the blood and what stays behind, right? What gets filtered out in the glomerulus? So you got things that go into the filtrate. Whoops. And things that stay behind. Right? Not everything gets pushed out of the blood into that capsular space. But generally, things that are smaller than 3 nanometers, that's about the cutoff. If something is smaller than 3 nanometers, it can get through the feet of that podocyte and out of the blood into the filtrate. So things like water, right? Glucose, small molecules, individual amino acids, right? Just individual free amino acids can get filtered out. Pretty much any small lipophilic molecule. That's what gets filtered out. Things that stay behind. I already mentioned it, but I'll write it down now. Cells, right? Those are definitely bigger than 3 nanometers. Proteins, whole proteins. This is why if you get a urine analysis done and they see lots of protein in your urine, that's a problem. That's a big problem because under normal working conditions, proteins are way too big to get filtered out. So if you see proteins in somebody's urine, then that means that there's something wrong with the glomerulus, right? Or somewhere there's something wrong because proteins should not be able to make it past the podocytes. So if you look here at this stuff that's getting filtered out, right? This is important stuff. Some of it's really important stuff. Water, got to have that, right? Glucose, got to have that. Amino acids, again, got to have that. A lot of small lipophilic molecules that your body needs to hold on to as well. So that's why so much of the energy that is expended by the kidneys is spent reabsorbing a lot of this stuff in the proximal and distal convoluted tubules. So if we look at the proximal and distal convoluted tubules, specifically the reabsorption that's going on there, most of it's happening in the proximal convoluted tubule. And there are 4 different ways it can happen. One is primary active transport, right? That's really the simplest way to think about it, And there's really only one thing that gets reabsorbed by primary active transport. Sodium. That's it. It's the only thing that's reabsorbed back into the blood via primary active transport is sodium. Everything else either diffuses on its own or, in the case of water, via osmosis. Or secondary active transport using the glucose gradient or the sodium gradient as a driver. So that's the second way, is secondary active transport. So things like glucose, vitamins, amino acids, ions that are not sodium. The list goes on and on. All that stuff gets absorbed by secondary active transport using sodium gradient as the driver. So this will sound familiar if you look at the next slide. The next slide looks a whole lot like an enterocyte. So you remember when we were looking at the cells of the small intestine and how things like sodium and, how things like glucose and other carbohydrates, proteins, nucleic acids, all that stuff gets absorbed from your small intestine via secondary active transport using sodium as the driver. It works exactly the same way in the kidneys. So if you look at that image on the next slide with the proximal convoluted tubule epithelial cell, it's basically, it looks a little bit convoluted because it got all those different activities going on one slide. But really all that's happening is the sodium-potassium pump pumps sodium into the blood from the cell, and so that creates a gradient just like it did in the enterocytes. And so there are co-transporters that use that gradient to move sodium into the cell with other solutes like glucose, amino acids, ions, vitamins. They all come in by secondary active transport. Some lipid-soluble substances can come in via diffusion, right? Just simple diffusion. If they're lipophilic, they can cross the plasma membrane. This is why drugs that are very lipophilic tend to stick around in your system for a long time, because they can't effectively be concentrated in the urine, because when they get concentrated in the urine, they just diffuse out and diffuse back into the blood. And then the fourth way things can get reabsorbed is osmosis. Right? Water can pass through aquaporins into the cell and then back into the blood. And water will just follow the solutes, right? If primary active transport is pumping sodium out of the tubule and back into the blood, then water will just go with it via osmosis, because it's going to go, if the concentration of sodium goes up over here, then water is going to go over there as well. So as you pump sodium, water will just follow it via osmosis through aquaporins. So yeah, most of this reabsorption happens in the proximal convoluted tubule. And when I say reabsorption, that's just short for reabsorption from the convoluted tubules into the peritubular capillaries, which are the blood vessels that surround the convoluted tubules. So on the next slide, what you'll see is the convoluted tubules. You can see the convoluted tubule branching off of the renal corpuscle, and as it kind of worms its way around through the cortex, it's surrounded by a tuft of blood vessels called the peritubular capillary. So that peritubular capillary just surrounds all those convoluted tubules. So when all this stuff leaves the convoluted tubule and goes back into the blood, it's going back into the peritubular capillaries. So the flip side of that tubular, the flip side of the reabsorption coin is tubular secretion. So at the same time you have all that stuff being resorbed from the tubules back into the capillaries, at the same time that's happening, there's also stuff that's being actively secreted into the tubules, out of the blood. So tubular secretion is, the nephron's way of actively pumping things out of the blood and into the convoluted tubules. So things that your body needs to get rid of, like waste products. Right? Like urea, uric acid. These are just waste products of amino acid metabolism. We'll talk a little bit more about urea later on in the chapter. This is also your body's way of regulating the pH of your blood. So if the pH of your blood gets too low, right, what that means is that the concentration of hydrogen ions is too high. Right? pH in your blood is low. It means your blood is too acidic. There's too much hydrogen ions there. So the cells of the convoluted tubules will actively pump hydrogen into the tubule out of the blood. So that will bring the pH of the blood back up, right? Make the blood less acidic. So it can work the other way too. It can pump out carbonate ions. If the blood is too basic, then the tubules will secrete hydrogen into the tubule out of the blood. If the blood is too basic, it will secrete carbonate ions, bring the pH back down. And all that's regulated by hormones, which we're actually not going to get to that this semester. All right, so that's tubular secretion, and just, I didn't write it down, but you should write it down. It's secretion from the blood into convoluted tubules. This is the stuff that the kidney is getting rid of, actively getting rid of it. I'm going to take a break because I need to plug in my camera. Go wash your hands. And we're back. Okay, so after the fluid leaves the proximal convoluted tubule, then it passes into the loop of Henle. So we're just going to draw the loop like this. We're not going to draw the convoluted tubules. So you've got fluid coming in this way and then leaving that way. The fluid comes in here, goes down, then turns and comes back up. So there are two parts to the loop of Henle. There is a descending limb of the loop of Henle, and there is an ascending limb. So in the descending limb, the filtrate is descending down into the lower areas of the medulla, And in the ascending limb, the fluid is coming back up. Right? Makes sense. That's why it's named what it is. And before we look at how this is all working, because this might be one of the most challenging subjects to explain and to really grasp in all of A&P 1 and 2, is how exactly the loop of Henle sets up a concentration gradient. So before we go into that, I'm just going to tell you like the nuts and bolts of what's going on here. The ascending limb is water permeable, right? What that means is that the descending limb. The descending limb is water permeable. What that means is that the cells that line the descending limb have lots of aquaporin, so water can get in and out very easily. The ascending limb, on the other hand, is not water permeable. All right? No water can get in or out of the ascending limb because there's no aquaporins. And in the ascending limb, sodium is actively pumped out of the loop and into the interstitial space. So yeah, in the ascending limb of the loop of Henle, sodium's actively being pumped out of the loop and into this interstitial tissue, right? Not necessarily the blood, but the tissue here around the loop. It's just being pumped out of the loop. And so if you look at what's happening here, the effect is something that we call a countercurrent multiplier. So just follow what's happening here. All right, the ascending limb pumps sodium out of the loop actively. So sodium's actively being pumped out of the loop here. And what that does is it raises the concentration of sodium outside the loop of Henle. Right? So when you raise the concentration of sodium outside the loop of Henle, that's going to pull water out of the descending limb. So water gets pulled out of the descending limb, and so as the fluid travels down the descending limb, it gets more and more and more concentrated. And then it crosses over into the ascending limb, and then comes back up, and it gets more and more and more dilute because sodium's getting pumped out. And so since these are running, well, they're running counter-current to one another. That's why we call it a counter-current multiplier. And down here at the bottom, because the descending limb is water permeable, at the bottom of the loop, the fluid inside the descending limb and just this interstitial fluid here, it's all isotonic, right? There's the same concentration of sodium in the loop here as there is outside. But as you go down, it gets more and more and more and more concentrated, right? So what you end up with, right, the punchline here, the thing that I want you to remember is that because of the way this is set up, I mean, I also want you to remember this stuff too, right? But the punchline here is that at the very bottom of the loop of Henle, what you got is a very high concentration of sodium. So if you look at your next slide, what you're looking at is just a very simplified view of the loop of Henle, and then the proximal and distal convoluted tubules, and then the collecting duct. And so you can see as you go down deeper into the medulla, the osmotic gradient gets higher and higher and higher. And all that means is just, think of that as the sodium concentration in the interstitial space, right? In this area right here. So the sodium concentration is getting higher and higher and higher and higher and higher as you move down into the deeper areas of the medulla, because the loop of Henle is creating that high concentration of sodium down here. So the osmolarity, the osmotic gradient, gets higher and higher and higher as you move down into the medulla. So when you look at the deeper areas of the medulla, right, the deeper areas of those renal pyramids, what you see is an extremely salty area, right? A tissue that has a really high concentration of sodium. And so I've actually got, when I was trying to figure out a way to explain this a few years ago when I put the course together, I put together an Excel sheet model, which I can give to you, but it's kind of, I need to be there to explain it with you. It's weird. So I'll give that to you. I think it's actually already on D2L. But really, it's just a model that, it models those three things that are happening, because there's only three things happening here. You've got sodium being pumped out of the ascending limb, water being pulled out of the descending limb, and then the fluid moving along with it. So take a look at that model, see if you can figure it out. It's hard to explain if I'm not there with you. But if you can't figure it out, don't worry about it. What I really want you to know here is what's happening at each point, right? What's happening in the descending limb, what's happening in the ascending limb, and what's the overall effect, right? The overall effect is this countercurrent multiplier that produces a really salty tissue in the deep areas of the medulla at the bottom of the loop of Henle. And that's one thing, I can't stress this enough. You know, it's a big misconception that the loop of Henle is just creating this high concentration of salt within the loop. And that's the case, but that's not the end of the story, right? Because the function of the loop of Henle is not to create a concentrated filtrate in the loop. The function of the loop of Henle is to create a very salty area down here in the interstitial fluid of the medulla. So if you look at that slide that we're still on right now, right, that has the osmotic gradient and the loop of Henle coming back up into the distal tubule that empties into the collecting duct, right? Everybody look at that PowerPoint slide. Look at where the distal tubule empties into the collecting duct. From there, the fluid will go down through the collecting duct, and that collecting duct goes down through that really salty tissue of the medulla. And that's where the concentration actually finally happens, because as the fluid passes down through the medulla, through that collecting duct, and it passes into that really salty tissue, all the water gets pulled out, right? So it's the collecting duct where the final concentration of urine actually happens. And I'm not going to write that on the board, but if I did, put big fat stars next to it, right? That's a huge point. The final concentration of urine happens in the collecting duct, because when the collecting duct passes down through that really salty medulla tissue, the water gets pulled out through aquaporins. And so you can imagine if you had a longer loop of Henle, you could make that tissue even saltier. So organisms that have really long loops of Henle, they're able to concentrate their urine really well, right? If you have a longer loop, then you have more space to create that gradient. So a long loop lets you create a better gradient, a stronger gradient in those deep areas of the medulla. And if you have a stronger gradient, then that means that you can pull more water out of the collecting duct. And so there's one organism on the next PowerPoint slide called kangaroo rat. It's adorable. Doesn't ever have to drink a drop of water its entire life. Lives in the desert. It's good, right? Because there's not a lot of water in the desert. It actually gets all of its water from, water is a byproduct of cellular respiration. And the kangaroo rat's kidneys are so efficient because it has really long loops of Henle. Kangaroo rat kidneys are so efficient that it can concentrate the urine so much that it can retain more water from cellular respiration than it needs to eliminate all this stuff, right? So that's why kangaroo rats never have to drink water. Cool story. All right. I told you we'd come back to talk about urea. We'll talk about urea. Urea is just a waste product of amino acid metabolism. It's, your body's way of getting rid of all that nitrogen waste. So it's a waste product of amino acid metabolism, looks like this. There you go. That's urea. So it's just a way to get rid of this stuff. And it's also used by the kidneys to amplify the osmotic gradient in the medulla. So here's how that works. You got your loop of Henle. Oop, try again. You got your loop of Henle. I'm just going to draw the distal convoluted tubule. It's not going to be very convoluted at all. Just for simplicity, there's your distal convoluted tubule. And then the collecting duct, right? So this is the descending loop of Henle. This is the ascending loop of Henle. There's your distal convoluted tubule right here, and then collecting duct. Okay, so filtrate is going down distal loop of Henle, comes up the ascending loop of Henle. In the distal convoluted tubule, urea is actively secreted into the distal convoluted tubule. Remember we talked about that earlier? This is where it happens. So urea is actively pumped into the distal convoluted tubule, right? By primary active transport. And then that fluid goes into the collecting duct. And as the fluid moves down the collecting duct and water gets pulled out, the concentration of urea gets higher and higher and higher. So the urea gets concentrated as it moves down the collecting duct. Now here, the kidneys actually do a neat little switcheroo trick with urea to amplify that osmotic gradient. because as you get down into the lower areas of the collecting duct, you enter an area where the collecting duct is permeable to urea. And so urea, trying to juggle markers here, urea can diffuse out down here, and it does diffuse out down here by quite a bit because the concentration here in the collecting duct is so high. So this is facilitated diffusion. And this part of the ascending limb is also permeable to urea. So urea gets pulled out there and then passes on into the ascending limb where it just gets recycled. So what happens here, you can follow the sequence of events. Cap up all my stuff. All right, in the distal convoluted tubule, urea gets actively secreted into the tubule, and then the urea gets concentrated as it moves down the collecting duct until it gets down here where the collecting duct is all of a sudden permeable to urea. So then the urea will diffuse out by facilitated diffusion and then diffuse on into the ascending limb of the loop of Henle, again by facilitated diffusion, because there's such a high concentration here and there's a low concentration here because the urea hasn't been pumped in yet. So here the urea is just following its concentration gradient out of the collecting duct into the interstitial fluid and then into the ascending limb of the loop of Henle, where it then goes up and on again. So when urea passes out of the collecting duct into the interstitial fluid, that's going to raise the osmolarity of the interstitial fluid because urea is a solute, it's a water-soluble molecule. And the more water-soluble molecules you have here, the higher the osmolarity. And so the higher the osmotic gradient. So that's going to pull even more water out of the collecting duct, and it's going to concentrate the urine even more. But when urea passes into the ascending limb of the loop of Henle, it's not going to bring water with it because water can't get in the ascending limb. See, water can follow the urea out of the collecting duct, but water cannot follow urea into the ascending limb. So this is just going to pull even more water out of that collecting duct just to amplify that urine concentration. So this is how urea is used to amplify the concentration of urine. One last time before the urine passes out of the kidney. All right, we'll talk about 4 different ways urine formation can be regulated. Regulation of urine formation. There's 2 steps that can be altered when the kidneys are regulating urine formation. One is at the filtration step, right? At the glomerulus. If more fluid gets pushed out of the glomerulus, then your kidneys make more urine, right? Makes sense. And then the other way that urine formation is regulated is the concentration step. So these are at the collecting duct, when water is being pulled out of the collecting duct, that's another step that can be altered. But those are the only two ways that urine formation is regulated. One is at the filtration step, and then the other is at the concentration step. So we'll look at filtration first. So we're looking at ways that the glomerular filtration rate can be altered. The GFR. So the first mechanism for regulating the glomerular filtration rate is called the myogenic mechanism. All right? And this is, this is a function of one of the properties of smooth muscle, where if you stretch out smooth muscle, how does it respond? It responds by contracting, right? If you stretch out a smooth muscle cell, then that's going to open up those stretch-gated receptors, or those stretch-gated ion channels. They stretch out, let a bunch of sodium in, starts an action potential, they contract, right? So that's how smooth muscle responds when you stretch it out. So look at it this way. If your blood pressure goes up, right, what's that going to do to the glomerular filtration rate? What do you reckon? It's going to increase the glomerular filtration rate, right? Makes sense. If you've got, fluid that you're trying to push through a filter, you raise the pressure of that fluid, more fluid is going to get through the filter, right? So if the blood pressure goes up, then your kidneys are going to start filtering faster for obvious reasons. But if your blood pressure goes up, that's also going to stretch the afferent arteriole, right? It's going to stretch all the blood vessels, but in particular, the afferent arteriole is going to be stretched. So the afferent arteriole gets stretched, and what does smooth muscle do when you stretch it out? It constricts. So that leads to the constriction of the smooth muscle there. And so if the afferent arteriole going into the glomerulus constricts, that will slow down the glomerular filtration rate. Again, makes sense. So the blood pressure goes up, that'll speed up the glomerular filtration rate, but it'll also stretch the afferent arteriole, which will cause a constriction of that smooth muscle in there, which will slow the glomerular filtration rate back down. It's just a negative feedback loop, right? So that way, your blood pressure, you don't get these wild changes in glomerular filtration rate every time your blood pressure goes up. Like if you're constantly stressed out and your blood pressure is really high, you're not just peeing all the time, right? Because there's a negative feedback loop that keeps glomerular filtration rate, right there in the window where it needs to be. So that's the myogenic mechanism. Next one is called tubuloglomerular feedback. Tubuloglomerular feedback. So this relies on a structure called the macula densa, which is on your next slide. No, it's not. It's on a slide after that. There we go. So I'm looking at the slide that has the glomerulus with the afferent and efferent arterioles, and then a structure called the macula densa labeled. And so what you need to in order to understand this mechanism, you need to understand that when the proximal convoluted tubule, so you look up there in that top left image with the whole nephron, right? When the proximal convoluted tubule leaves the renal corpuscle, it'll go and do the convoluted tubule thing and then go down into the loop of Henle, up the ascending limb of the loop of Henle to the distal convoluted tubule, and then it'll make a pass back by the renal corpuscle where it originated. And so if you look back at the video now away from the PowerPoint, say my hand, my fist right here is the renal corpuscle, and you've got an afferent arteriole and an efferent arteriole, and they're just kind of coming out of the same side of the renal corpuscle. So here's my renal corpuscle, afferent arteriole, efferent arteriole. The distal convoluted tubule sits right there at the crux of those two arterioles. So the distal convoluted tubule, when it comes back up out of the loop of Henle, it'll pass between those two arterioles, right next to the renal corpuscle. And there's a clump of cells right there between the distal convoluted tubule and between those arterioles called the macula densa. And the macula densa can detect and respond to sodium concentrations. So I'll show you how this works. Here's what happens. Let's say for whatever reason there's an increase in the glomerular filtration rate. I don't know, maybe your heart is pumping faster. It could be a number of things, right? Either way, there's an increase in the glomerular filtration rate. So more fluid is being produced. And what happens is if the filtration rate picks up, then the fluid is going to move through the nephron a lot faster. And if the fluid is moving through that nephron really fast, there's less time for reabsorption. So there's less time for sodium to get pulled out of the filtrate and back into the blood. So when fluid gets pushed out of the glomerulus, right, all the sodium goes with it. And if the fluid moves through the nephron really quick, then by the time the fluid gets back to the macula densa, there's been less time for resorption. So that means that there's a higher concentration of sodium at the macula densa. So an increase in GFR gives you less time for reabsorption. Right? And if there's less time for reabsorption because the fluid is moving through the nephron a lot faster, that means that you're going to have a higher concentration of sodium at the macula densa. And so the macula densa will respond to that high concentration of sodium by releasing vasoconstrictor paracrine molecules. So the macula densa will release all these vasoconstrictor paracrine molecules. Remember, a paracrine molecule is just a molecule that can act on a close by cell, make it do something. In this case, cause vasoconstriction. So the afferent arteriole will constrict. So the afferent arteriole going into that glomerulus will constrict in response to those vasoconstrictor molecules, and that will slow down our glomerular filtration rate. So just recap, because it was kind of broken, right? GFR picks up, filtration rate picks up, so there's more fluid moving through the nephron. The fluid's moving through the nephron a lot faster, so there's less time for reabsorption. That means there's a higher concentration of sodium in the filtrate when it gets to the macula densa. The macula densa responds to that by releasing vasoconstrictor paracrine molecules, and those paracrine molecules cause the afferent arteriole to constrict, which lowers the glomerular filtration rate. And the third way GFR is regulated is through the renin, angiotensin, aldosterone mechanism. So if you got a drop in blood pressure, this time we're looking at a drop in blood pressure that it counteracts. Drop in blood pressure causes the release of renin, well, the activation of renin, which, I mean, you know, the renin, angiotensin, aldosterone pathway. We talked about that in chapter 15, so I'll just draw it like this. Renin activates angiotensin, which then causes the release of aldosterone, and aldosterone causes water retention. So your body retains more water, that's going to bring blood pressure back up. Which will counteract the effect of the drop in blood pressure, which was, oh wait, did that wrong. Don't worry, I'll recap here in a minute. All right, recap. Drop in blood pressure causes a slowdown of the glomerular filtration rate. So your kidneys aren't filtering as fast, right? So drop in blood pressure slows down the GFR, but drop in blood pressure also causes activation of renin. Renin converts angiotensinogen into angiotensin. Angiotensin causes the release of aldosterone. Aldosterone causes water retention. If your body's retaining more water, then the blood pressure goes up. And if your blood pressure goes up, that's going to bring up the GFR. So this is just another negative feedback loop. These are all negative feedback loops. All right, so those are three ways that GFR is regulated. The other way to regulate urine formation is at the back end, right, at the concentration step. And if you recall, concentration, the final concentration is happening where? It's not happening in the loop of Henle. It's happening in the collecting ducts. So, just to recap, before I jump into this, you need to understand how urine is concentrated. When the urine is passing down through the collecting duct, the collecting duct goes down into that really salty tissue of the medulla, water gets pulled out of the collecting duct and into the interstitial fluid, right? Everybody with me? Okay. So how does water cross out of the collecting duct into the interstitial fluid? Right? Because remember, water is polar. Water is by definition water soluble, right? So it can't cross the plasma membrane on its own. But to get out of the collecting duct, it's got to cross two plasma membranes, right? It's got to get into the collecting duct epithelium, and then it's got to get out of the collecting duct epithelium. And it does that not just by crossing the plasma membrane, it does that through aquaporins. That's what I want you to understand before we go into this. The water that's getting pulled out of the collecting duct is passing through aquaporins. If there were no aquaporins there, then the water can't get out of the collecting duct. So the way that concentration is regulated is by regulating the expression of aquaporins in the collecting duct. So, let's say you got a drop in blood pressure, right? That's going to cause the release of antidiuretic hormone from the pituitary. And antidiuretic hormone, if you remember, it's in the name, antidiuretic. It inhibits diuresis, urine formation. So antidiuretic hormone is going to inhibit urine formation, or slow down urine formation. So what it actually does is it causes the production and expression of aquaporins in the collecting duct. Production and expression of aquaporins in the collecting duct. And so that makes the collecting duct more water permeable. And if the collecting duct is more water permeable, that means more water gets pulled out of the collecting duct, which means that you retain more water. And if you retain more water, that's going to bring your blood pressure back up. It's going to bring your blood pressure back up. Right? Recap. Drop in blood pressure causes ADH, antidiuretic hormone, to be released from the pituitary. Antidiuretic hormone causes the production and expression of aquaporins in the collecting duct. So that means the collecting duct is more water permeable, which means more water gets pulled out of the collecting duct, which means your urine is more concentrated. You hold on to more water, brings your blood pressure back up, or it keeps it from going down any farther. Right? And remember, this is that hormone that gets inhibited by alcohol. This is why you feel hungover after getting drunk, because you're dehydrated, right? Because the alcohol inhibits the activity of antidiuretic hormone, so the collecting ducts don't produce aquaporins. That means the water just passes all the way down through the collecting duct without getting pulled out, and so you just start peeing copious amounts of water when you start drinking a whole lot. So that's why you get dehydrated. Okay, so that's pretty much the end of kidney physiology. The rest of this chapter is devoted to where the urine goes from there. So, if we look at our renal pyramid again, right? You got all these collecting ducts. Actually, I'm going to make this better. I can do better than that. Yeah, that's good enough. Okay. So you got collecting ducts. I'm only going to draw two collecting ducts. but there are lots of collecting ducts in each renal pyramid. The collecting ducts come together into these ducts that go down into the renal papilla, which is this area right here. So, you know what? The reason that I keep staring at my notes is because there's a method to my madness, right? We'll try this again. Here's your pyramid. We'll start over. Here's your renal pyramid. The renal pyramid empties into a calyx. All right, so here's that open space at the tip of the renal pyramid called a calyx. All right, this part of the renal pyramid right here that extends down into the calyx is called the papilla. This is one of those things you got to identify for the lab exam. Yeah, so this part of the renal pyramid is called the papilla. All right, so now we can talk about collecting ducts. Here's a collecting duct, right? Here's another collecting duct. There's more than just two, but we'll just draw two. They come together down here in the papilla and enter into a duct called a papillary duct. So there are ducts that extend down through the renal papilla called papillary ducts, and they just kind of collect all the fluid coming from multiple collecting ducts, and it's the final duct before you end up in a calyx. So these are the collecting ducts, and these are papillary ducts. I don't know why that bothers me so damn much, but it does. Do like that. There we go. That's a little bit more clear, I guess. I don't know. Yeah. So if you look at the flow of urine, right, from the nephron, you go from the urine is produced as a filtrate at the glomerulus, and then so the fluid gets pushed out of the glomerulus into the renal corpuscle, and then from there it goes into the proximal convoluted tubule, and then from there it goes into the loop of Henle, and then from there it goes into the distal convoluted tubule, and from there it goes into the collecting ducts. And from there it goes into the papillary ducts. From there it'll go into a calyx. And then from the calyx it will go into the pelvis. And from the pelvis it will go into the ureter. So that's the whole chain of events, right? We're not done yet. You got to see it all the way out. We'll back up and look at our full gross schematic of the urinary system. Right? These are your kidneys. The ureters, exit the medial side of the kidneys and then go down. So there's a ureter. And the ureters empty into the urinary bladder. And so the ureters empty into the urinary bladder through an orifice, a hole called the ureteric orifice. All right, the ureteric orifice is just the hole in the urinary bladder through which the ureters empty. And then urine exits the urinary bladder through the urethra, which is just the duct carries urine from the urinary bladder out of your body. And the last slide all right, wait, no, the next to last slide. No, this is the last slide. Yeah. Okay. This last slide is just a cross-section of the urinary bladder, so you can see the structures that kind of control it, right? There's a lot of smooth muscle in the urinary bladder. And that smooth muscle is called the detrusor muscle. You can see the ureters coming in from either side. Can't see the ureteric orifices in this image. But yeah, that smooth muscle that surrounds the urinary bladder is called the detrusor muscle. And where the bladder kind of comes to a narrow neck, it's just called the bladder neck. And then that opens up into the duct that carries urine out of your body, which is the urethra. And kind of like with the anal canal, there's two sphincters. There's an external urethral sphincter and an internal urethral sphincter. And the internal urethral sphincter is just smooth muscle right there in the neck of the bladder. And the external urethral sphincter is skeletal muscle, so you actually control that one. And it really functions kind of similarly to defecation, because it's reflexive, right? Just like with defecation, but the impetus for this reflex is not distension of the urinary bladder. It's actually relaxation of the external sphincter, which is skeletal muscle. So when you relax the external sphincter, that causes a reflex that causes contraction of the detrusor muscle to push all that urine out. So that's why when you go to pee, you just relax, and then the bladder responds by contracting and pushing all the urine out. And I think that's it. Yep. All right. Go wash your hands again.